It's a great pleasure to introduce you to Dr. Marianne Anderson. She is a clinician scientist at the Institute, a clinical haematologist at the Victorian Comprehensive Cancer Centre. She's been very fortunate. She's been involved in the early clinical trials, and it'll be very interesting for me to hear from her speaking as a clinician scientist at the very beginning of your career and what you've learned from this. Thank you, Marianne.
Hi, so my name is Marianne Anderson, and clearly I'm a little bit more junior than some of the talks that have happened already today. And that's because I started on this journey back in 2011 when I commenced my PhD. And I think one of the things that I've really taken away from this really remarkable journey is that as a doctor coming into the lab, you realise that you don't know much yourself, and you really need a lot of help from a variety of different people to get up to speed in science and laboratory techniques, and really to understand what's going on. You need to collaborate with a whole lot of different groups around the Institute, and also sort of outside of the Institute in the precinct, and as we've heard today, internationally. So I'll talk to you today about my journey since 2011, when I started 6 months before the clinical trials of our BCL2 inhibitor began. So we had, when I started my PhD, we had a new class of drug that was designed to selectively target BCL2. This drug is now approved for clinical use, but when I started my PhD, no human being had ever actually had this drug. And really the journey of my PhD was the journey of treating the patients together with Andrew Roberts and seeing how the drug worked in patients and treating them. And also it was a journey of trying to understand in the laboratory the ways in which the drug was working and trying to predict how patients would respond to it and which patients were likely to do well and which patients perhaps not so much. And very excitingly, this drug was then approved in April 2016 by the US FDA, and now just a month ago it was approved in many other countries, but also in Australia. And we've discovered that in certain subgroups of patients with previously treated CLL, this drug can actually, these patients, we would not expect them to have a very long prognosis. They're patients who have disease that we would expect to have killed them. And these patients have been brought many years of extra quality life from this drug. And I think that really highlights what all the work over many, many years by all of the speakers in this session has achieved. And really, it is the basic research that made this possible. So the concept of targeting BCL2, as you heard today, is not new. And it's been attempted by others in a variety of different ways. But there were many failures. And I think that also highlights the fact that when you're starting to do something that is entirely new, you have to be prepared for failure and you have to keep trying. And, you know, I think once you have a drug that works and you're seeing the outcomes in patients, it really does make all of that effort worthwhile. And drug development really is a testament to the power of great science, but also to wholehearted collaborations. No one person or one specialty field can achieve this. It does require so many different talented individuals from so many different walks of life. So as we've heard today, the drug that I worked on during my PhD was the outcome of a really fruitful collaboration between WEHI and Australian academia, but also between US pharmaceuticals, Genentech and AbbVie. But the research scientists who were involved include the biologists, the chemists, the structural biologists, but also clinician scientists and other members of the clinical teams. And I've got some photos here of some people who really were predecessors to me and taught me a lot of what I know. So my PhD supervisors, Andrew Roberts and David Wang, and then 3 other haematologists who are all good friends and colleagues who were in the lab prior to me, and I learnt from them and took on the baton, so to speak. And then this photo over here is the team that we worked with in the clinic at Royal Melbourne and Peter Mac to give patients this drug and to collect the data and to keep them safe. So we now have a new class of drug, and this class of drug can achieve something that many others have tried to do with CLL, but many others have failed. And what you can see here is a blood film of a patient with CLL, and you can see that these small purple dots, so to speak, are an excess number of lymphocytes. And in some cases, when these lymphocytes accumulate too much, the patients become sick and unfortunately in some cases die. And we have a drug now that within a matter of hours can turn a patient's blood film from this to this, and that's an almost entirely normal blood film. And for us, that's very, very exciting. And I think that's what most scientists are hoping to achieve in their career and to work towards, to be able to actually make a difference to people. So we have a drug that is a new class. It's specific and it addresses an unmet need for patients. This is only the beginning, however, and I'll be handing the baton over to other up-and-comers, no doubt. And really what we're trying to do now is to work out in further clinical trials, and there are more than 30 ongoing trials in Australia and also around the world, trying to work out exactly where this drug fits into our armamentarium, what combinations will work best, which patients to target, and it's going to be a very exciting area of study for many years to come. And there are ongoing translational studies, which myself and my supervisors are involved in, really looking to see both blue sky projects, but also applied projects. And this is really supported by both philanthropy, government, and collaborations with industry. And I know myself that my PhD work wouldn't have been possible without the Webster bequest, which paid for it. And so all of these groups coming together are really achieving something that I think is quite remarkable, and I hope that you agree. Thank you.